talk about linearization of genome sequence graph. What is linearization? We want to order and orient all the nodes in the graph. So we have to define a linear order on the set of nodes, and every node has to be oriented. That means that one of its strands will be the main strand, or alternatively, every node has two sides, and one of its sides will be called inside, and the other side will be called outside. On this picture, the left-hand side is inside and right-hand side is outside. When the graph is linearized, all its edges are classified into three categories: forward arcs, feedback arcs, and reversing joins. Forward arcs are edges that are compatible with both orientation and order. That is, there are those solid edges on this picture. So every forward arc leads from an outside of some node to an inside of another node, and the outside node precedes the inside node in the order. Feedback arc are compatible with orientation but incompatible with order, so those are the edges that are pointing backwards. And finally, reversing joins are the edges that join two outsides or two insides, and those cannot be oriented. And what is a good linearization? We consider three metrics. First two are the sums of weights of reversing joins and feedback arcs, respectively. And the third one is the average cut width. Here we consider cuts of the graph that go between two consecutive nodes, between every pair of two consecutive nodes, consecutive in the order. And cut width is the number of edges that are crossed by the cut. So in this example, the consecutive cuts have widths 2, 3, 2, 2, and 1. So the average cut width is 2. The first two metrics are motivated by the fact that we generally want to have the graph as similar to the directed graph as possible. And the third metric is motivated by the fact that even if we have a large number of genomes, we usually expect that the number of different alleles at a particular locus is moderate, and the cut width seems to be related to the number of these alleles in particular positions. There is one previous work that I know about this problem. It was presented a few years ago on RECOMB. The authors proposed a two-step approach. In the first step, the nodes are oriented, and in the second step, the nodes are ordered. After orienting the nodes, all the reversing joins are established. So in the second step, the graph could be interpreted as a directed graph, and the authors considered two approaches. One was based on a heuristic to the feedback arc set problem. That is, feedback arc set is to order the nodes of a directed graph such that the number of feedback arcs will be minimized. It is an NP-hard problem, and there are several heuristics that solve this problem, and they use one of these heuristics. And also they propose a novel algorithm that focused on minimizing the average cut width. Both these algorithms are implemented and included in a variation graph toolbox. And we wanted to investigate deeper this area, in particular to analyze the reversing joins and design an algorithm that would minimize both feedback arcs and reversing joins. So if you want to minimize feedback arcs and reversing joins, so then we should maximize forward arcs. So the idea is to add edges to, to, to the graph from the heaviest to the lightest and try to make them forward arcs, to establish as many as possible forward arcs. And at each step of the algorithm, all the nodes are oriented and ordered. Ordered are separately on each connected component. And when we add edges, we can modify the orientation and order, but in the way that won't change the classification of the edges that are already in the graph. And there are several cases. First, if we add the edges that join two separate connected components, there are two cases. Either our edge joins an outside of some node in one component with the inside of a node in another component. And in that case, we can simply add this edge and join the orders of two components in the way that the nodes from the first one precede all the nodes from the second one. Then the edge is classified as a forward arc. The second case is when the edge joins two outsides or possibly two insides. In that case, we can reverse the orientation and order in one of the components, and then we will have the previous case. The edge will join an outside with an inside, and we join these components in the same way. When we add edge to that both ends has in the same component, there are a few more cases. Two of them are simple. If the edge joins two in or two outside, that it must be classified as a reversing join. Otherwise, if the edge joins an outside with an inside and the outside node precedes the inside, then it can be classified as forward arc, then we classify it as a forward arc. And if neither of these conditions is satisfied, it means that the edge joins outside with the inside, but the inside node precedes the outside nodes, like in this picture, as the inside node precedes the outside node. So this edge can be classified as a feedback arc, but it may be also, but sometimes it may be also classified as a forward arc. To decide whether this is the case, we can check whether there is a path from the inside node to the outside node that consists of the forward arcs. If there exists such a path, then this edge must be classified as a feedback arc, because if we to this path add this edge, it will form a cycle, and a cycle cannot be cannot consist of the forward arcs only. And if there is no such a path, then we can classify this edge as a forward arc, but we have to modify the order of the nodes. And we to make this modification, we first identify all the nodes within this interval between these two ends of the edge, all the nodes that are reachable from the inside node of this edge. These are all the nodes that are reachable by forward arcs and lie within this interval. Those are these green nodes. This is the first group, and then we identify the nodes from which the outside node is reachable. These are the red nodes, and we modify the order in the way that all the red nodes precede the green nodes first, and and moreover the order within the green nodes is the same as before. It is saved, and similarly, the order within the red nodes is also saved. And we modify the positions in the order of only of those red and gray nodes, red and green nodes, and all the other nodes save their positions in the order. So this is these are the main ideas of the algorithm. And to make it efficiently, we need to carefully design the data structure. And our data structure is based on the data structure for this joint set, the classic data structure, also known as a union find structure. And those and set of nodes represent connected components, and moreover we have additional attributes that that hold information necessary to determine the order and orientation of the nodes. And these attributes could be simply defined for every node, but in that case, we would have to modify in some operation, we would have to modify those attributes in many nodes. Instead of it, we introduced some attributes that define the orientation and the position in the order relatively to the parent of a node in a tree of the union find structure. And then we could, when we want to reverse the whole connected component, reverse or change the order, we can only modify the parameters in the root of such a tree, which makes some of these operations of adding the edges much more efficient. To evaluate our structure, we prepared simulated and real data sets. This is in a simulated data, we tried to reproduce the data that was used in the paper of Hausler et al. In real data, we used genomes of Escherichia coli bacteria, and we took from 10 to 150 genomes, and for each genome set, we created graphs using the variation graph toolbox. And here are the results on the simulated data. As we can see, our algorithm ALIBI performs best in sense of weighted reversing joins and weighted feedback arcs, while in the sense of average cut width, the best is the flow procedure of Hausler et al. And on the real data, the results are similar. However, the differences in reversing joins and in feedback arcs are, especially for larger number of graphs, are much higher. They have around the 3 order of magnitudes. Note that here is the logarithmic scale on this graph. So there are the difference is around 1000. And moreover, in the sense of average cut width, the flow procedure performs excellent up to 50 genomes, but then it's getting much worse. Sometimes it's even worse than the other algorithm. Here are the complexity results. As we can see, our algorithm is has the lowest memory and is faster than two other algorithms, and the difference is getting higher for larger number of genomes. So to conclude, our algorithm alibi substantially outperforms its competitors in terms of weighted reversing join and weighted feedback arcs, and is fastest and has the lowest memory requirements. But in the sense of average cut width, the flow procedure achieves best results. We work on improving alibi in terms of average cut width, and some preliminary results are promising. It seems that after applying small modification, we can have average cut width similar to the flow procedure, and the other parameters won't significantly change. And I hope it will be implemented and submitted to GitHub in a few weeks. Thank you for your attention.
Sorry about that. But I have two devices connected at the same time. Thank you for the talk. There is one question from the audience. It's rather a long one, but I will read it in parts. Thank you for the talk, Norbert. Maybe I misinterpreted the problem definition, but how different is the idea of linearization to that of a bidirected De Bruijn graph?
Sorry, how different is
the question
idea of
goes
linearization?
My understanding is that linearization is an attempt to Dagify regular genome graphs, for instance, the Bruijn graph. If this is true, can we view linearization as decomposition of a graph into a linear multiple sequence alignment?
Yes, the linearization the linearization can be applied to De Bruijn, to bidirected De Bruijn graphs. It can be applied to any kind of bidirected graphs. I don't know what do you mean by Dagifying.
Yeah, makes sense. I think at this point it could be fruitful that you reply to this question using the chat and question and answers, and maybe the attendee who was anonymous can follow up, and we'll continue offline then.
Generally, I would say that linearization is then Dagifying the graph is the optimal situation. This is the situation when we will obtain no feedback arc and no reversing join. Then we will obtain a DAG. however, it is not always possible, and so our algorithm tries to minimize the number of feedback arcs and reversing joins.
Okay, thank you. Since we are out of time, I think we'll take no more questions. And that was the last talk of the session. We have a short break, I believe 10 minutes, and we start with the last session of the day.
Okay. Thanks to all the speaker and thanks to Ryan for chairing the session. I shared the last postcard here. Okay, and